Welcome all to this video lecture. This is John Milton, working as an assistant professor in the Department of Microbiology, St.Joseph College of Arts and Science, Autonomous in Cuddalore. From the subject of medical virology, I will going to uh, take a lecture, that is adenoviruses. So the taxonomical information of the adenoviruses are they are belong to the family of Adenoviridae and genus mast adenoviruses. They are first detected in human in the adenoid tissues. So the adenoid tissues are nothing but it is the tissues that is found just above the vocal cord. This is the vocal above the vocal cord. The tissue will be present, that is called as adenoid tissues. From this region, the adenoviruses are first isolated and first detected. The mast adenoviruses, which infect both human being and animals. The human adenoviruses are divided into 6 subgenera designated as from A to F, A, B, C, D, F. There are 6 subgenera are there in the human adenoviruses, and these subgroups contains 47 serotypes. They are numbered from 1 to 47, 1, 2, 3, 4, 5, 6, like up to 47. The morphology of the adenoviruses are their size. The size are medium sized, that is about 70 nanometer to 90 nanometer in diameter, and they are spherical in shape, and they possess the double stranded DNA as a genetic material, nucleic acids, and they also having the envelope. They are the type of enveloped viruses, and the structure about the capsid is composed of 252 ranged icosahedral capsomere symmetries. The capsomeres are nothing but it is the protein subunit of the capsid. The icosahedron has 20 triangular facets and 12 vertices. So this is the structure of the adenoviruses. Of the 252 capsomere, 240 are hexons. Hexon means they having 6 capsomere, adjacent capsomeres, and 12 pentons. Penton means these having the 5 adjacent capsomere units. From each penton project an apical fiber with a knob at the tree and giving the virus appearance of the space vehicle. You can see the viruses they're having the tree-like structure on each penton basis, and that gives the appearance of the satellite. The cultivation. Regarding cultivation, the adenoviruses are highly host specific. That means a particular adenoviruses always infect a particular host, that is called as a host specific. The viruses which affect the human being cannot affect other kind of organisms, that is the host specific, and can be able to grow in the tissue culture of human origin only, because all the viruses which infect human being can only grow in the human cell line and human organs. We cannot cultivate these adenoviruses in the other egg embryonic cell culture and other cultures. We cannot cultivate the organisms. So the cell line which are used for cultivating these viruses is HeLa and HEP2, and they also can use the embryonic kidney of human being for cultivating this organism. The cytopathic changes consist of rounding and aggregation of the swollen cells into grape-like cluster. The cytopathic changes is nothing but after entering this organism, these viruses into the cytoplasm of the host cells, that will make the nucleuses into disintegrated form and giving the appearance of the grape-like cluster like this, like this. So that is called a cytopathic effect. The intracellular inclusion can also be seen when we do the staining from this preparation, viral infected tissue preparation. The resistant to physical and chemical agents. The viruses are heat labile. Heat labile means they are sensitive to the heat. The heat can degrade or disintegrate or inactivate the virus. That is the meaning for the heat labile, and can be destroyed in minutes at 56 degrees Celsius. At 56 degrees Celsius, when we keep these viruses, that will be killed. So that is a highly heat labile. The heat will denature, will inactivate, will kill the viruses immediately. Especially when we expose around 56 degrees Celsius for 1 minute is enough to kill the viruses. They are relatively stable up to 37 degrees Celsius for 1 week. When we maintain the temperature around 37 degrees Celsius, that is the body temperature of us, the viruses can be able to survive for 1 week, up to 1 week. They are resistant to other ether and bile salt. Ether and bile salt having the viruses are resistant to these two antimicrobial agents. Pathogenesis. The adenoviruses are transmitted by direct contact. Direct contact is the route by which the viruses can be transmitted from one person to another person, and also the fecal-oral route. Fecal-oral route means the fecal contaminated food and water that will be consumed by the ingestion through the oral route, that is through the mouth, that is called as a fecal-oral route, and sometimes through the water. Consumption of the water also one of the reason for causing the infection. Sometimes lead to persistent asymptomatic infections in the tonsils, adenoids, and intestines. Okay, sometimes the viruses which create persistent asymptomatic infections. Persistent means for a long period. Asymptomatic infection means it does not showing any symptoms, especially in the region of tonsils, adenoids, and intestines. The adenoviruses cause infection of the eye, respiratory tract, urinary tract, and the gastrointestinal tract. These regions can be infected with adenoviruses. The infections may occur through conjunctiva or nasal mucosa. Conjunctiva means that is the layer of over the eye. Through this, the infections may occur, organisms may enter, that is the meaning. And nasal mucosa. Nasal mucosa means the mucosal layer which surrounds the nasal region. In children, it may occur through fecal-oral route, that is the food and water which is contaminated with the fecal matter of infected persons, and when we consume this food and water that will cause infection, that is the fecal-oral route, and the incubation period is 6 to 7 days. The viruses multiply in the conjunctiva or pharynx or small intestine and then spread to the lymph nodes. The clinical symptoms includes the lymph nodes are which are specialized cells that will having most number of macrophages and other cells which are involved in the phagocytosis that has important role in the in fighting against the pathogenic organisms. The clinical symptoms include pharyngitis and tonsillitis. The pharyngitis and tonsillitis will be caused by the serotype 1 to 7, 1, 2, 3, 4, 5, 6, 7, up to 7. The pneumonia that is caused by the serotype 3 and 7. And acute respiratory diseases which is caused by the serotype 4, 7, and 21. And pharyngoconjunctival fever that will be caused by serotype 3, 7, and 14. Epidemic rectoconjunctivitis that is caused by serotype 8, and follicular conjunctivitis caused by serotype 3, 4, and 11. Acute hemorrhagic cystitis is caused by 11 and 21 serotypes. Infantile gastroenteritis caused by serotype 40 and 41. So laboratory diagnosis, how can we diagnose the disease by the laboratory techniques? Adenoviruses infection can be diagnosed in the laboratory by antigen detections. The presence of the antigens which is produced by the viruses will be detected in the laboratories. That is one of the technique, and we can also employ the PCR technique and as well as the virus can be isolated by using the cell line HeLa and Kep2 and other human kidney cells like this. And also the serological test also used to diagnose the presence of these viruses and the infections. The specimen which are taken for isolating the viruses and finding the viruses viral presence are throat swab, conjunctival swab, anal swab, urine, genital secretion, bronchial lavage, transtracheal aspirate, nasopharyngeal aspirate, autopsy and biopsy materials. So these are all the sampling materials for diagnosing the adenoviral infections. Microscopy. So by using the microscopy, we can also check the presence of the viruses in the fecal matter. The fecal specimens can be taken out, and from the fecal specimen, fecal matter, we can prepare the specimen, and the specimen can be seen by the electron microscopy for the presence of these viruses. The immunofluorescent technique, that is also one of the immunological techniques. So here the viral antigen in the cells from the respiratory tract, eye, urine, autopsy and biopsy materials can be found out. An infected cell culture by immunofluorescence using monoclonal antibodies. The monoclonal antibodies can also be used to check the presence of the particular antigens in the infected cell cultures. Isolation. The isolation of the viruses in the tissue culture and identification by the cytopathic effect, complement fixation, immunofluorescent test. These are also other techniques. The viruses can also be isolated, and based upon the cytopathic effects, we can confirm the virus presence, the infections. The serology. The serological diagnosis includes a demonstration of the increase in titer of the antibodies in the paired sera. So the increase in antibodies also one of the indication of these viral infections. The next adenovirus is SV40. The adenovirus grown in monkey kidney cells may get contaminated with SV40. If we grow this some adenoviruses in the kidney cells that may be contaminated with the SV40 viruses. Both the viruses multiply without producing cytopathic effect on the cells, on the kidney cells. When the infectious SV40 is removed by using specific FE40 anti-serum, the leftover adenovirus exhibit tumorigenic properties. Usually the SV40 viruses is the viruses that will be grow when already the adenoviruses are present in a cell culture. If you remove the SV40 cells from the cell culture, the adenoviruses are more toxic than the previous one that has the property of a tumorigenic in nature. Tumorigenic means that can cause the cancer. The virus particle may contain the genome of SV40 and the capsid of adenoviruses. This has been called adenovirus SV40 hybrid. So this is regarding the adenoviruses. Thank you for watching this video lecture.